Gene cloning. Cloning means produce multiple copies. So gene cloning actually is produce multiple copies of a specific genes. The purpose of gene cloning is to produce an organism with new genotype, and the new genotype determines the desired phenotypes that we need to observe for this species. And the most common example is how the characteristic of an eukaryotic organisms such as human can be introduced into an example of prokaryotic cell, which is bacteria. Through gene cloning, the specific copies of genes can be produced, and the copies of genes can be isolated from the cytoplasm of the bacterial cell and used for other specific purposes such as research purpose, environmental purpose, and agricultural purpose. Other than this, the bacterial cell, which is the whole cell, will express the genes, which means they're able to produce the products encoded by the genes, which is the proteins. And the proteins can be harvested or extracted from the cytoplasm, purified, and commercialized. The first step is isolation of DNA. So the DNA materials will be separated from the origin of cell. There are two types of DNA will be isolated during the gene cloning. The first is target DNA. Target DNA contains gene of interest. That means this segment of DNA contains the genes which is required for certain purpose. The most common topic to be discussed is the human insulin production. Pancreas is the human organ that able to produce insulin, and insulin is the hormone able to lower down our blood glucose level to normal level. So human pancreatic cell is the target cell that carry the target DNA which contains gene of interest. Is a segment of the DNA that coded for insulin production. The second type of DNA will be isolated is plasmid, the small, circular, and double-stranded DNA isolated from bacterial cell. And the example of bacteria, which is E. coli, will be used throughout the process of gene cloning. Plasmid will be used as the cloning vector in gene cloning, and it has selectable genetic marker, which is LATZ gene and ampicillin resistance gene. After the isolation, both DNA will be cleaved or cut by restriction enzyme. For example, the target DNA will be cut by EcoR1. And usually it has more than one restriction site can be recognized by the EcoR1. After cutting, the target DNA will be cut into a few restriction fragments, but only one restriction fragment carried the gene of interest. For example, the gene for insulin. Plasmid will be cut by the same restriction enzyme, which is EcoR1. The purpose of cutting with same restriction enzyme is to produce the complementary sticky ends. So usually plasmid has one restriction site for specific restriction enzyme, which is located at the LATZ gene. Both restriction fragments and linear plasmid, they have the complementary sticky ends. So when they are mixed under an optimal condition, they allow the hydrogen bond formation. But a recombinant plasmid is completely made when the DNA ligase catalyzes the joining of the linear plasmid with the foreign gene through the phosphodiester bond formation. So here are the examples of a recombinant plasmid that carried the foreign gene, and some of it carried the foreign gene contains the gene of interest. However, there are some plasmid they do not have enough time or they do not have the chance to get close to the restriction fragments. So the sticky ends will pair up together and form an original plasmid. I would like to start with the transformation, which is a process when the recombinant DNA or recombinant plasmid will be inserted into the host cell, which are actually the bacteria or the example E. coli that used in the first step during isolation. Even though the plasmid is considered small circular DNA, but it still have difficulty to be reinserted back into the bacterial cell. So we need to increase the membrane permeability to the plasmid. So the method is started with the cold calcium chloride treatment and followed by a heating process. So at the end of the transformation, the host cells able to carry the recombinant DNA, or some of it will carry the original plasmid. Basically, there are three types of host cells are produced at the end of the transformation. The bacterial cell containing the recombinant plasmid, that means the plasmid is inserted with foreign gene, the gene of other species, or the bacteria contains the plasmid which is original, or we call it as non-recombinant plasmid. But some of the bacteria, they are unable to carry or unable to be inserted with any plasmid. And these types of bacteria cannot be recognized by naked eyes. We have to identify the colonies through a screening step. Let's study amplification in step 4. Amplification means make many copies. So to produce many copies of recombinant plasmid, we need to provide a medium for the bacteria to grow into a large number. And the large number of bacteria we call it as colony. So scientists will prepare a solid nutrients medium which contains the antibiotic ampicillin and also the nutrients for the bacteria to grow, sugar agar. So here is a simulation how the bacteria can be transferred from the solution into the agar plate, and then we culture it in an incubator. After the incubation, we can observe the colonies are formed at the surface of the agar plate, even with naked eyes. But why we have two colors of colonies, which are blue and white? It's because the last step of gene cloning is screening, which is also called as blue-white screening. This step is very important to help us differentiate the bacterial colonies with recombinant plasmid from the bacterial colonies without the recombinant plasmid. And the blue colony and white colony forms are actually due to the presence of LAT-Z gene. So from the amplification in step 4, we will notice that there are the formation of white colony and blue colonies. Okay, these are formed due to the bacteria, no matter which bacteria they are, is either containing the recombinant plasmid or non-recombinant plasmid. As long as the plasmid is found in the cytoplasm of the bacterial cell, they carry the gene of ampicillin resistance. So the bacteria will not be killed by the ampicillin. They are able to survive in the agar plate and reproduce to form colonies. And always remember, there are some bacteria they are unable to carry any plasmid during the process transformation. So without plasmid in the cytoplasm of the bacterial cell, this bacteria will be killed by the ampicillin. So they won't form any colony in the agar plate. And now with the formation of white colonies and blue colonies, we move on to the detail or the concept behind it, how we can identify the bacteria with the recombinant plasmid. I would like to start with blue colonies, which contains the bacterial cells carrying non-recombinant plasmid, or we say the plasmid still in original form. So, if we look at this plasmid which is in the original, that means the Z gene still intact. Maksudnya masih sama, tidak ada insertion yang lain. So, the Z gene we say it is functional because it still can undergo the process transcription to produce the mRNA, and the mRNA will be translated to produce the protein, which is actually the enzyme beta-galactosidase. This enzyme will enable the bacterial cell to hydrolyze the S-gal in the medium plate. So, when the S-gal sugar has been hydrolyzed by the enzyme, it can form the blue color product. And the blue color product will color each of the bacterial cell into blue color. This is why we have blue colonies formed. White colonies consist of bacteria that carrying recombinant plasmid. The recombinant plasmid can be the plasmid containing the foreign gene that contains the gene of interest, or the recombinant plasmid can carry the foreign gene without the gene of interest. No matter which type of recombinant plasmid they are, as long as they has been inserted into the cloning vector, they have disrupted the Z gene. Or the Z gene is non-functional because it cannot be transcribed successfully by RNA polymerase. So, no mRNA production, no translation, no protein encoded by Z gene can be produced. The protein is actually the enzyme beta-galactosidase. So without the enzyme, the bacteria cannot hydrolyze the S-gal sugar in the medium plate, so no blue color pigment will be produced. But the bacteria still can grow by using the other nutrients found in the medium plate, and through the binary fission process, from one bacterium, they form many copies of bacteria, even though they are colorless, but because of the large number of these bacterial cells, they form white colonies in the agar plate. So, at the end of the gene cloning, scientists will focus on white colonies because white colonies has the bacteria containing recombinant plasmid. But always remember, the recombinant plasmid may contain the gene of interest or without gene of interest. So how we differentiate the white colonies with the gene of interest, which is our target colonies? So just consider as an extra knowledge to differentiate the white colonies with gene of interest from the white colonies without the gene of interest. We have to move on to one more step, which is nucleic acid hybridization. But with the development of the molecular technologies, we have another way which is more efficient. We call it as complementary DNA technology. This technology will still give us white and blue colonies, but the difference is now the white colonies all contains the gene of interest. This is because the first step, we are going to isolate the mRNA from the target cell. It's not more DNA isolation. And mRNA consists of the exons for the target gene only. So same as the complementary DNA technology or cDNA technology, you are going to study one more technology in second year, which is the PCR, Polymerase Chain Reaction. Okay, polymerase chain reaction is very useful to amplify the DNA molecules into a large quantity, so we can do more investigation on other purpose. But now, why we have to practice home-based learning or distant learning is because of the COVID-19. And the positive cases can be detected by many ways. One of the more accurate way is actually the PCR testing. So here is just a simple sharing to you how the PCR testing can be used to test the positive cases. You are going to study this PCR method more detailed in second year. So here